Hello, this is Gina Piscatelli with our last lecture for week 4 in Anatomy and Physiology 1. In this lecture, I will discuss how proteins and lipids are used as organic nutrients or fuel to make ATP. Before we begin, let's just look at the four major steps, I should say, of ATP production through aerobic cellular respiration. So the first step is glycolysis. That happens in the cytoplasm. The second step is pyruvate oxidation, and that occurs in the intermembrane space of the mitochondria. And the third step is called the Krebs cycle, or the citric acid cycle. It happens in the matrix of the mitochondria. And then last and certainly not least is the electron transport chain, which occurs due to the fact that there are proteins embedded in the inner membrane space that are pumps and ions. We get the majority of ATP production from the electron transport chain because ATP synthase is present in this chain and it can produce a lot of ATP. So far in our discussions, we've only talked about using glucose, which is a carbohydrate, as an organic nutrient. So our bodies eat complex carbohydrates, and our digestive system breaks down the complex carbohydrates into small molecules that we call monomers, and specifically in carbohydrates, monosaccharides. These monosaccharides begin the process of aerobic cellular respiration. First glycolysis, then the transition reaction or pyruvate oxidation, then the Krebs cycle, and then eventually, the electron transport chain, which isn't shown on this graph. But what about when we eat a lot of protein or fat, lipid? Our cells can use that material. In fact, even stores in our body can be broken down. Stores of protein, stores of lipids can be broken down for our cells to use as starting material to make ATP. Now, the material does not get turned into ATP, but the starting material will be broken down, release energy, and that energy will be captured to fuel the production of ATP. So as you know, proteins are made up of monomers called amino acids, and I'm just going to deal with triglycerides. Triglycerides are made up of a backbone of glycerol and fatty acid chains. So we're going to see that the process for proteins is different than for lipids or for carbohydrates. First, the amino acids have to have the nitrogen taken off, the amine groups, so that's called deamination. But then the resulting product can usually enter somewhere in the Krebs cycle, somewhere in here. Now for lipids, it's a little bit more complicated because we have two types of compounds making up a triglyceride. A glycerol is a carbohydrate that has 3 carbons, so it's going to enter somewhere at the end of glycolysis. Fatty acids are chains, long chains of carbon and hydrogen that need to be cut up, and that process is called beta oxidation. And the products of beta oxidation are 2 carbon compounds that can be converted to acetyl-CoA. So they will enter the Krebs cycle. So first let's look at protein catabolism. When proteins are broken down through the process of hydrolysis, that gives us amino acids. Now those amino acids have nitrogen and a carboxyl group attached, but the nitrogen is a waste product for us, so they must first be or toxic to us in high concentrations. So they must first be deaminated. And the process of deaminating Sorry, computer got stuck. The process of deaminating a protein involves enzymes, and we're not going to study all the different enzymes, but essentially this amine group has to come off. Now it's going to come off and it'll travel in the blood and it'll eventually go to the kidney, where the kidney will convert it into urea, which is something that goes into urine, and we will get rid of it. It'll be lost from the body. But what's left over is an amino acid without its amine group, so it's not really an amino acid anymore. But notice that this particular amino acid has 5 carbons. Okay, it also has 2 carboxyl groups, but it has 5 carbons, so it will mimic one of the components or intermediaries in the Krebs cycle. So I'm going to show you this crazy Krebs cycle picture for just a second and look at 5 carbons. Hello, this is Gina Piscatelli recording your last lecture for week 4 in Anatomy and Physiology 1. In this lecture, I will talk about how proteins and lipids can be broken down by the body to produce energy that will be harnessed to make ATP. Before we begin, let's look at the 4 major steps involved in aerobic respiration that produces ATP. So the first step is glycolysis, that occurs in the cytoplasm. The second step happens in the intermembrane space, and that's called pyruvate oxidation. The third step occurs in the matrix of the mitochondria, and it's called the Krebs cycle. And then the last step we call the electron transport chain. There's more than one thing happening here, but that's at the intermembrane, intermitochondrial membrane. And it involves the pumping of hydrogens as well as the production of ATP as those hydrogens diffuse down their gradient. So we've got 4 major steps in 4 different locations. And so far what we've talked about are carbohydrates as the organic nutrient that's broken down to give us energy to make ATP. When we eat complex carbohydrates that are broken down by our digestive system into small monomers called monosaccharides, such as glucose. So we looked at glucose going through glycolysis, pyruvate oxidation, the Krebs cycle, and the electron transport chain, not shown on this diagram. Today, instead, what we're going to do is look at how proteins, and specifically triglycerides, not all fats, just triglycerides, how they enter this aerobic cellular respiration pathway. So we'll see that proteins are broken down into the monomers called amino acids, which will eventually enter the Krebs cycle. Triglycerides have 2 different kinds of monomers, sort of. One is a glycerol backbone, and that's a carbohydrate, so it's going to enter at the end of glycolysis. It's got 3 carbons. And fatty acids are really long chains of carbon. They have to be cut up, and they are then converted, these pieces are converted to acetyl-CoA. So I would say that triglyceride fatty acids really enter the Krebs cycle. So bypassing glycolysis for the most part. So first we'll look at protein breakdown. So protein catabolism. When a protein is hydrolyzed, individual amino acids are the result or products. This happens in our digestive tract, and then the amino acids are taken up into our cells. They can also be taken up into the blood and travel all over your body so that many cells can take them up. Before they can enter the chemical reactions of aerobic cellular respiration, the nitrogen has to be removed. The nitrogen, you'll remember, is in an amine group, so amino acids are first deaminated. So deamination will require an enzyme to break off this amine group. We aren't going to study those really, but that amine group will then enter your bloodstream, go to the kidneys, and the kidneys will filter out the urea and allow from the blood and put it into our urine, allow it to leave the body. It'll be excreted. But what we have left over is the backbone, I guess, of the rest of the amino acid right here, just no amine group. This one you'll notice, because I started with a certain amino acid, this one has 5 carbons. It closely resembles one of the intermediaries in the Krebs cycle. And the one that it closely resembles is alpha-ketoglutarate. So I'm going to show you a picture of this crazy Krebs cycle thing. And if you look at the step down here that has alpha-ketoglutarate, 5 carbons, that is this molecule. And there's 2 carboxyl groups on the end, and there's 5 carbons. So that's very similar to the amino acid that I just showed you. And I'll go back right here. So here's a carboxyl group, here's a carboxyl group, and there's 5 carbons with 1 double bound to oxygen. It's the same. It's alpha-ketoglutarate. Okay, so that will enter the Krebs cycle. There are other amino acids with different numbers of carbons that enter the Krebs cycle as well. Like aspartate, for example, is very similar to oxaloacetate, which has 4 carbons. Alanine is the one that's a little strange because it's only got 3 carbons. It enters at the transition reaction. But in general, just proteins start at the Krebs cycle because that's where amino acids enter. Now let's talk about triglycerides a little bit. Now, triglyceride structure we should review. This is the glycerol backbone. So when lipolysis occurs or hydrolysis of a triglyceride, the fatty acid chains are removed from that glycerol. It has 3 carbons. So it's going to mimic a molecule somewhere towards the end of glycolysis, maybe even pyruvate. And then the fatty acid chains are long chains of carbon, right? They're too big to really enter at any particular stage that we've talked about so far. Well, this fatty acid's kind of short, but usually they're quite long. So we need to talk about what's the fate of these fatty acids. They've been broken off. What happens to them? They go under a chemical process known as beta oxidation. And there are enzymes that do this. And what they do is cleave the fatty acid chain after every 2 carbons. That's where beta comes from. So alpha would be first, and then the beta carbon. So the number 1 carbon is alpha, the number 2 carbon is the beta. And so it'll be cut here, then it'll be cut here, and so on and so forth. So you can see that this is the fatty acid chain that we started with that has 12 carbons. Whoops, that doesn't look good at all. That has 12 carbons. And the product after beta oxidation is a fatty acid chain that only has 10. The process will happen again and again and again. So 2 more will come off, and it'll come 2 more, and then 2 more, and then 2 more. And so one fatty acid chain produces several acetyl-CoAs, and therefore it acetyl-CoA will go directly to the Krebs cycle. I guess I could just circle it, the Krebs cycle. So from one particular fatty acid, you can turn the Krebs cycle many times, produce lots of NADH, and ultimately, run the electron transport chain a lot. So triglycerides with their 3 long fatty acid chains really produce a lot of ATP. Okay, that's the bottom line for cell metabolism. No more cell metabolism for you. Thank you very much.